A pleasant evening to one and all present here. I once again welcome all of you who have joined us for today's session. Today we are starting with the new series called GRASP 2024. We'll be starting with biochemistry. It is a very useful series for all the exam going first years. And we are very excited to have Professor Dr. Desigamani Kanniyappan sir, PhD, Department of Biochemistry at Meenakshi Medical College, Kanchipuram. Welcome, sir. We are very honored to have you. To give the opening remarks, I invite Dr. Raghunandan Ramanan.
Very good evening to all. It is my pleasure to welcome my faculty, my mentor, Dr. Desigamani sir, with whom I have spent almost my first and second year. Still, he was in my college, one of the fantastic faculty, and still I am a student, and still I am learning many things from him. And thank you so much, sir, for joining with us today. And this is going to be the wonderful beginning for the first years, I guess. And this year we are planning extensive sessions by different faculties and a complete revision series which is planned by Parthipan, and I wish him good wishes for him for organizing such a wonderful session. And today it will be a really a wonderful session, and you are going to learn something that you haven't so far. So it's not like just you're going to learn about one particular topic in biochemistry. Basically, you're going to learn how to approach biochemistry and how to approach any topic. So this is going to be an eye-opener for everyone. And thank you once again, Parthiban, for organizing this wonderful session in this channel. And now I'd like to request Dr. Jamuna ma'am, our faculty advisor, to give her opening remarks.
So good evening all. At the outset, I congratulate Team Medicine for organizing this GRASP 24 series. And I'm really glad that our opening batsman with the IPL season, Dr. Desigamani sir, a wonderful biochemistry teacher, and he really makes a complicated biochemistry look very, very simple for you. And all his points, all are take-home messages for the first years as well as for preparing those preparing the NEET examination. So we are really honored to have Dr. Desigamani sir with us. And I request all the students to make use of all the sessions in this series so that you can really excel first year with so much of confidence and with good marks. So all the very best, and over to Dr. Desigamani sir.
Good evening. Thank you, madam. Dr. Jamuna madam and Dr. Raghunandan for the opening remarks and a very good words about me, and Dr. Parthiban, young buddy doctor. Hopefully, I will do justice as you have narrated about me. Thank you for the opportunity, madam. Okay. Dear learners, can you hear me? Audio is good, no, ma'am?
Yes, sir. Yes, sir. Yes, sir.
Thank you. Without wasting our time, let's start. As the topic says, amino acid metabolism. I would start with a simple question. What is amino acid? Why a medical student supposed to learn the amino acids? What is amino acids? It is one of the major nutrients, or it is one of the major nutrients. As you all aware, there are major nutrients like carbohydrate, proteins, and lipids. Okay. And then micro minerals or minor minerals like minor nutrients like vitamins and minerals. What's the importance of this major nutrients, amino acids? We'll just learn about that. Before that, how do you differentiate amino acid from the rest of the major nutrients like carbohydrate and lipids? That is also most important. Why? Because when you learn the structural aspect of the nutrients, which will tell you all the physical and chemical properties of the nutrients. Okay. First of all, I wanted to give you, why do we have to learn these nutrients? Whether it is today's topic, amino acids or carbohydrate or lipids. First of all, you should know, why do we have to learn the nutrients in biochemistry first year subject? The answer to this question, all our cells is made up of nutrients, please make it very clear. All our cells, which means different types of cell, 250 different types of cell, 40 trillion cells, all the cell, including all the parts of the cell, is made up of carbohydrate, proteins and lipids, some minerals and some vitamins is what is making the cell. So now it's very clear why we have to learn the nutrients, because they make our cells. And then the second aspect, as I told you, two important aspects, why we have to learn the nutrients. First one is very clear. Second one, that you study in physiology, the functions of cell. All the cells functions through the nutrients. So basically, our nutrients make the cell, each and every parts of the cell. Also the functions is carried by nutrients. So there is no other go without learning nutrients. You cannot understand what we are made of, how we are functioning. Okay, that's the reason I started by saying this. Now, how do you differentiate? How do you differentiate nutrients? How do you differentiate nutrients? Nutrients like amino acid from carbohydrate and protein. That's very important. Okay, I will just tell you how you can say this molecule belongs to amino acids. All amino acids. Okay, you must be knowing there are 20 different amino acids. 10 of them are essential, 10 of them are non-essential. Be it any amino acids, you should look for one single carbon having amino group as well as carboxylic group. If any molecule has got this kind of arrangement, blindly you can say this is called amino acids. Okay. If you know the answer for carbohydrate and lipids, please post it in the group. Okay. Why do we need to learn? Again, why do we need to learn? As I told you, these macromolecules and micromolecules make the cell, makes the cell, and then functions of cell is carried out by these nutrients only, Okay. We cannot spend a lot of time in understanding what are the molecules from which nutrients is involved in which cell type in making the cell. Okay. It doesn't make any sense. But of course, as a concept, please understand the nutrient that you take is going to make a cell. Which cell? All types of cell. If you are not getting the point, let me spend 1 minute of time and then make you realize the importance of this statement. Okay. From the day one of birth till you die. From the day one of birth till you die. What is going into a body? First day of birth, mother's milk is fed. Look at the mother's milk. It is given for nearly 2 years. Okay. Some doctors strictly prescribe without even water, only breast milk for 2 years. At least 1 year or 1.5 years, 2 years, depends on the setup. Okay. Later on, the babies are fed with whatever parents are taking. So now you need to look back. What are the events occurring when you give the food like mother's milk or the food that we take? What are the changes occurring in the body? As you can see, the growth of the babies is happening. That means, what can you explain? So possibly and probably there are nutrients in the milk which serves to build the cells in the growing baby. Same time, you also see the babies are alive. They are doing the function. They are surviving. How do you explain this? The same nutrient, one side making all the cell and the component to grow the baby. At the same time, the nutrients that we fed is only the reason for functioning of all the cells. Through that, the baby or we are surviving. Please make it this very clearly. This is why we learn biochemistry in the first year. If you look at the topics in biochemistry, major nutrients and minor nutrients. You have to learn the carbohydrate, you have to learn the protein, you have to learn the lipid, you have to learn the minerals, all minerals, and then you have to learn the vitamins, then water, then oxygen. This is what we are taking in. This is what we are taking in. So we are learning why? Because all these 7 substances, how they contribute in making the cell or functioning the cell. Hence, we can learn a life at a molecular level. Other topics, whatever you learn, ask me the reason, like enzymes and hormones and all that. They are all made in the body, enzymes and hormones. Why? To manage these molecules like carbohydrate, proteins, and lipids, how and when, how to use it, when this goes little excess, how to manage the excess, and then how to store it. This is the reason we learn all the topics that are there in the biochemistry. Okay. If you get the gist of this introduction, let me start. Amino acids. Why? As a medical student, we have to learn the amino acids. Amino acids is an important nutrient. Important nutrients. Are we consuming amino acid through our diet? No, it's not possible. We are not consuming amino acid through our diet. Why? Simple reason is it is not being cultivated as amino acids. Okay, it's not being cultivated as amino acid. The simplest answer, amino acid as it is not available in any source. Okay, instead it is available as protein. That's why we call dietary protein. What we are consuming is dietary protein. Why? Because it is amino acid form, it is not available. So whatever form is available, we are consuming. So dietary protein is taken. Dietary protein is taken. The dietary proteins are digested into its constituent amino acids. Okay. As you would have learned in digestion absorption, proteins are digested in the stomach by pepsin, also in the intestine with the help of exocrine gland secretes a lot of protein digestive enzymes. They are called proteases. They digest all types of protein and release oligopeptides and amino acids. Okay. Eventually, all the dietary protein becomes amino acids in the intestine before it gets absorbed. Okay. Now, please pay attention. These amino acids, now onwards, we call dietary amino acids. Dietary amino acids, though it was in the form of protein, but cells and body cells can recognize as amino acids only because you need to digest them into amino acids. Then the simplest form, amino acids can be absorbed. There are varieties of transporter mechanism, absorption mechanism, okay? Facilitated transport system or sodium dependence transport system. I'm not going to go detail. I'm laying down the concept. So there are a variety of transport system. Please do learn that because one or other person, if they don't express the transporter system, those amino acids will not even be absorbed. Accordingly, they will suffer. Okay. So it is essential to learn that. Through this transporter system, the amino acids from the lumen of intestine is getting absorbed by the endothelial cell or enterohepatic, epithelial cells in the intestine. Okay, it's also called enterocytes. Enterocytes, the apical surface which is in contact with the lumen, there you will have a transporter system. It gets absorbed. And then through the basolateral surface, another transporter system through which the amino acid escapes. Eventually, what you see there, the portal vein system. Okay, it enters into the portal vein system, reaches all into liver. All the amino acids, whatever is absorbed throughout the intestine, it goes to liver. Okay, whatever amino acids needed for the liver cell, they also take the amino acid through their own receptor system. But otherwise, all the other amino acids in the blood has to go to the systemic circulation. Okay. Essentially, whatever liver wants, they take. Rest everything is going through the systemic circulation. From systemic circulation, all your cells in the body, entire system will take their own need of amino acids. They also take through their own receptor system. Their own receptor system. What we have just seen from the diet, how these proteins are getting digested, how amino acids are released in the intestine, how they are absorbed and transported and reaching to all the cells. This is very fundamental. Okay. Now the million-dollar question is, what are the fates of these amino acids which are reaching into any cells in the body? I'll repeat. What are the different fates of these amino acids which are reaching into all the different cell types? In other words, why do we have to consume this dietary protein every day? 0.8 grams per kg of body weight. This is what is minimum prescribed amount. What is the reason? Or what is the benefit? Why we have to consume this protein? You must ask the question. Okay. Since it is not available in the amino acids, we have to consume through dietary protein. Dietary protein. What's the reason? Okay. You can post the answer in the group if you know. Otherwise, please listen carefully. There are very important vital roles played by these amino acids. We can have them in an hierarchy manner. Number one, you must be knowing there are more than lakhs of protein in the body at any given point in time. I'm not even mentioning which cell or which organ. Okay. Collectively, in any person at any given point in time, there are more than 1 lakh proteins are present. This is a statement. This is a fact. Okay. Why do we need this 1 lakh different proteins? If we don't have them, we will not survive. We cannot survive. Proteins are very important. These 1 lakh different proteins are made by the individual cell. Okay. Individual cell by using these dietary amino acids. Dietary amino acids. So basically, you provide the substrate for the cell to synthesize different types of protein, what they want at that point in time. Okay. All the organs needs the protein. Without protein, we will not survive. Remember, I told you two important aspects of the diet. One, which helps to make the cell and all the component of cell. Now ask me the question, whether the dietary protein or the dietary amino acid is going to contribute in making the cell. Absolutely, the answer is yes. Okay, you will see the cell membrane having the protein, mitochondrial membrane having the protein, lysosomal membrane having the protein. Any membrane, you will find presence of protein. Just not presence of protein, they do wonderful functions. To make it very simple, what functions the protein which are present in the cell membrane does it? Any water-soluble substance has to pass through the membrane only via proteins. If there is no protein, respected protein, the water-soluble molecules cannot pass through or cannot exist the cell. Entry and exit, water-soluble molecule needs the protein carrier or protein channels you would have learned. Okay, these all the proteins on the cell membrane, not on the peripheral membrane alone. Even any organelle, you must be aware, all organelles are also having double membrane or single membrane. Every membranes in the cell or cell organelles should or must have their own respective proteins. These are all proteins manufactured with the help of your dietary amino acids. Okay, not only that, as I told you, second important aspect, functions of cell, our diet helps. So now we are learning about amino acids. These amino acids contributes a lot of protein to be produced in individual cells. According to their need, whichever cell needs, whichever protein, they are produced at that point in time. If you are not appreciating this statement, I will give you an example. Glycolysis happens in all the cell with the help of what? Glycolytic enzyme. How many enzymes are there? Roughly about 10 different enzymes. What they are made of? They are all made of a protein. What do you think how they are produced? They are all produced like this only. Your dietary amino acids contributes in making the glycolytic enzymes. Likewise, There are thousands of different enzymes in the body. There are thousands of receptors in the cell. There are hundreds of hormones. They are protein in nature. Okay. Like you would have studied about the immunoglobulins. They are protein. Albumin, they are protein. Likewise, we can go on telling the molecules which are made up of proteins. Okay. So protein is manufactured or synthesized by individual cells according to their need. Whenever they are needed, they are produced. Okay. So what did you learn now? The dietary amino acids and its important functions. Why do we take the dietary amino acids through our diet? Why a doctor prescribes 0.8 grams of protein per kg body weight? Okay, this is the number one aspects. Number one aspects. Is it the only functions of dietary amino acids making a cellular protein? Not at all. What are the other aspects of helps by amino acids, dietary amino acids? There are molecules which are called peptides. Okay, don't get confused. Many textbooks says peptides and proteins, there is no differences like that. But you have to be very clear, at least for academic purpose. Okay, when I say peptides, they are lesser than 50 amino acid joined by peptide linkage. When I say protein, more than 50 amino acid joined by peptide linkage, more than 50 amino acid. One protein may have 10,000 amino acids also. No, don't worry. Minimum 50 and more than 50 number of amino acid joined by peptide linkage is called protein. It must have a specific function. Then it is qualified to call protein. Similarly, when I say peptide, biologically important peptides, lesser than 50 amino acid joined by peptide linkage is known as biologically important peptides. Why did I bring this second line, biologically important peptides? Because the dietary amino acid, first and foremost important function is to produce a lot of proteins for the cell. Second aspect, also helps to produce certain biologically important peptides. Okay, since there is no facility to converse with you, please post the answer in the group if you know at least one or two biologically important peptides which are produced by different amino acids. Okay, I will give you an example which is familiar to you all. Glutathione. Have you heard of glutathione? Glutathione, which is present exclusively in RBC. Okay, many cells do have, but I am taking an example of RBC, which has got high amount of glutathione. What they are made of? 3 different amino acids. Okay, glutamic acid, glycine, and cysteine. Okay, gamma, glutamyl, cysteine, glycine. Glutamic acid, cysteine, and glycine. These are the 3 amino acids which makes the peptide. Likewise, there are many peptides in many different cell types. Okay, just to give you some more example, I will not spend more time. Oxytocin is a peptide. Okay, biologically important peptide, nanopeptide, which means what? 9 different amino acids. Okay, most important note, what you have to understand, peptides are named after how many amino acids are there. If there is 3 amino acid joined by peptide linkage, you have to count only the number of amino acid, my dear. Tripeptide. If there is only 5 amino acid, pentapeptide. Okay. If there is 8 amino acid which makes one peptide molecule, octopeptide. 9 amino acid which makes the peptide, nanopeptide. Okay. Nanopeptide is what? Oxytocin. Okay. You know the functions of oxytocin. So what are you learning? Not only making the protein for an individual cell, also the dietary amino acid helps to produce, according to the cell, whatever biologically important peptides needed to be produced. Okay, that's why we insist have dietary protein, because it has to produce amino acid in the intestine and then go to the cell to help whatever molecules they need to be produced in terms of protein as well as biologically important peptides. Now ask me one more question. Okay. What is the fate of the protein? What is the fate of the biologically important peptides that are produced in the cell? Do you expect the protein? Please have the imagination. You have 40 trillion cells. Now, understand all your cells are receiving after you take the dietary protein into their cells. All the amino acid now in the cell, you see them, they are making into protein. Also, there is a production of biologically important peptide. Okay, don't assume all protein will be produced. Not at all. The rule of the cell is whatever protein is needed at that point in time, such protein only will be produced. Similarly, whatever biologically important peptides are needed, those biologically important peptides only will be produced. What next? Do these protein and biologically important peptide will remain in the cell forever? What is your answer? It cannot be like that. There are reasons. First, I will lay down the answer to you. As and when cell needs, those proteins will be manufactured or synthesized or made. After that, it is being allowed to function while it is produced. Once the functions are over, there are two very important systems inside all the cell. They take the protein and then they digest inside the cell to release its own amino acids. What will be the fate of amino acid which is released from the already used protein? It will be recycled. Whatever new needs, accordingly, these amino acids will be served for the same cell. I hope you got the point. I hope you got the point. For a sake, I'll repeat one more time. See, imagine the amino acid which is entering into cell, which produces a lot of protein as well as biologically important peptide. Both these molecule is allowed to function. Once the functions are over, it has to be degraded. It has to be degraded into constituent amino acids. So fantastic mechanism. If you have a question, where in the cell these degradation happens or digestion happens, intracellular digestion. One is lysosome. Another one is very new. Maybe most of you know it. It's called ubiquitin-related pathways. Ubiquitin. UBIQUITON. Ubiquitin. Ubiquitin-related pathways. I will just explain it very simple, briefly. The ubiquitin pathway. Ubiquitin is a small protein. Again, it's a small protein, 53 amino acid, I guess. Okay, 53 amino acid made up of one small protein, that's called ubiquitin. What it does, it just goes, identifies in the cell whichever protein is non-functional or old enough, not needed for the cell, just goes and tags those protein, tags it. Okay, once ubiquitin tags them, then it is surrounded by another proteolytic enzyme, proteasome. Proteasome is forming and then degraded into amino acids. Very simple. In the cytoplasm itself. On the other hand, the older protein might reach to lysosome. You all know lysosome is a suicidal bag. Why they called? Any molecule which enters into lysosome gets self-degraded. Okay. So in this case, what we are talking about protein and peptides, which can go to lysosome, there is an enzyme called, group of enzyme called cathepsin. Cathepsin, C-A-T-E-P-S-I-N, cathepsin. Those are the enzymes which are responsible for degrading the protein and releasing its own constituent amino acids. So what eventually you see, all the proteins are degraded in the cell intracellularly with two different pathways, releasing its constituent amino acids. Okay. Now, what we learned essentially, dietary protein or dietary amino acid. Please be clear about dietary protein and dietary amino acids. There are two lines I'm giving. Dietary protein, the protein which are present in any diet. Okay, someone may consume milk, someone may consume egg or fish or meat. Okay, so in those diet, whatever proteins are present, they are called dietary protein. So when you take it through your diet, till it reaches intestine, you may call dietary protein. Once in the intestine, when they are digested and release the amino acid, it becomes dietary amino acids. Please understand. Okay? Dietary amino acids. Now, is it the only two reasons why we take the dietary protein? What are the two reasons? They help to produce the cellular protein. They help to produce cellular biologically important compounds. Is it the only two reasons why we consume the dietary protein? The answer is no. There are still wonderful, important, life-saving functions. Okay. Please give an answer if you know. I can make a note. 6:30 is the time. Those who are typing, the time will appear. Then I will appreciate them after I see the message. What do you think? Apart from these two important aspects of dietary protein or dietary amino acid, the dietary amino acid is going to save our life. Okay. The third and the most important functions for dietary amino acid is to produce important compounds or important molecules. I'm not saying a protein. I'm not even saying a biologically important peptides. Now I'm making a demarcation. Important molecules. Important molecules. Sir, I'm not able to get the answer. What is this important molecules? Okay. Can you give some examples? Which important molecules? Which amino acid? In which cell it is produced? Likewise, can I think all amino acids in their own cell, they can give some important vital molecule through which we are functioning day-to-day basis? Absolutely, the answer is yes. Okay. I'm sure you would have heard neurotransmitters. Neurotransmitter. Most of them are monoamines. Okay. Example I'll give you. Dopamine, serotonin, epinephrine. I stop there. At least some 4 or 5 important neurotransmitter you should know. These neurotransmitters are produced by the cell, specific neurons. Take an example of dopamine, epinephrine. Okay, these are all produced by dopaminergic neurons in the brain. There are a bunch of fiber which set to release the dopamine. Okay, and then epinephrine and norepinephrine. Together they are called catecholamines. Okay, they are all produced with the help of tyrosine, a single amino acid called tyrosine. Please remember, tyrosine is also doing the first function, second function, and then the third function. What is first function? Tyrosine will be a part of several proteins in several cells. Remember that. Tyrosine also will be in some of the biologically important peptides somewhere in the peptide formation. Apart from these two major functions, tyrosine is also going into a cell, specific cell, and then helps to produce catecholamines. The same tyrosine enters into thyroid gland to produce thyroid hormone. What is thyroid hormone? Basically, tyrosine with iodine. 3 iodine on one single tyrosine, it becomes triiodothyronine, T3. Similarly, one single tyrosine, 4 molecules of iodine is called tetraiodothyronine or T4. Not only these two important functions, tyrosine can go to melanin pigment formations in the skin. Melanin is a compound produced from tyrosine. Likewise, there are many amino acids has got their own important product formations in different cells. Whichever cell needs a molecule, they take the respective amino acid and then start producing the product. Through that product, the cells are completing their functions. Please get that straight. Okay. Again, I'll make you this line. Why do we take the dietary protein? Why do we take the dietary amino acids? We never realized this. I'm sure as a medico first year, you would not have realized in your +2 levels. You never bothered to question what you're taking in the diet. How is it useful for your life? Okay. Please hereafter ask one question. Whatever you learn, first ask before learning why you learn this topic. Ask what you learn, why you learn first. Always ask this. If the answer is known to you, then it makes a purpose or meaning for you to learn. For you to learn. Okay? Without knowing the reason why you want to learn what you want to learn, I don't think you will get an interest. I don't think you will get an interest. Please ask this question. Why you learn what you learn, not only for biochemistry, any subject, any topic. Before you start learning, ask one question. Why I have to learn this? Where will I use this knowledge in my clinical practice? This is totally missing in students' life. I'm seeing everywhere. Students fail to realize why they learn what they learn. If you learn this, because I'm repeating several times, you realize it, how important it is to ask this question. If you know the answer for this, I am sure definitely unconditional love you develop on the subject or the topics, any topics. Okay. Again, I'll ask the question, why we learn amino acids? The answer is because we learn our dietary amino acid serves us to produce many different protein for many different cells. Without the protein, one example of protein, I gave you the glycolytic enzyme. Another example, insulin. You all know insulin as a hormone only, but basically it's a protein. If you don't produce protein, insulin, you are not going to metabolize your carbohydrate, proteins, and lipids together properly. Okay? That's the importance of learning the amino acid metabolism. Why we learn what we learn? Please ask this question. Again, why we learn the amino acid metabolism? When we learn, we come to know these dietary amino acid helps to produce many proteins in many cells. Secondly, these dietary amino acid helps to produce many biologically important peptides. Example, glutathione. What's the importance of glutathione? Now I ask you the question. What's the importance of glutathione? Can we not survive without glutathione? Absolutely, the answer is yes. You can't survive. Glutathione helps to protect the RBC membrane. If there is very drastic reduction in glutathione synthesis, RBC membrane cannot be protected. Hence, there is going to be an hemolysis. If the extent of hemolysis is going to be very severe, then you may lose all RBCs, and then oxygen supply to the organs will be compromised. Slowly, you may lose the potential functions, and then it may lead to death if you are not intervening and correcting. Similarly, there are a lot of peptides. Want of time, I am not giving all the names and their important functions. Please search and then learn different, at least 10 different biologically important peptides. Why did I use the word biologically important? For our human life. At least 10 different peptides, biologically important peptides in different organs or cell type. You learn the names of different peptides and their important functions. Then appreciate, oh my God, these peptides are produced through our own dietary amino acids. And then come to the third important aspect. Because you consume the dietary amino acid, now we learn there are many different life-saving compounds are produced. Life-saving compounds are produced in different cell type. One example I gave you. Tyrosine is an amino acid. Tyrosine is an amino acid. When you take them in your diet, we don't even know which diet tyrosine is present. Don't bother. You mix and match, or you take the good protein, so-called good protein. What is good protein? Good protein which one has got all the essential amino acid, including non-essential amino acids. Okay, all essential amino acid basically. Example is an egg and then milk. Gourmet food. Okay, you can afford to have. Then you get all the benefits. Take an example of tyrosine. Apart from making several proteins, several peptides, it is also producing wonderful life-saving molecules. Imagine if you don't produce dopamine, epinephrine, norepinephrine, what will become? You will become Parkinson's disease. You cannot have a gait, the postural position. You cannot walk properly. You cannot do your day-to-day activities. because of this simple molecules called catecholamines production lacking. Similarly, if you don't consume adequate tyrosine, not only not consuming, okay, I am going to lay down how these molecules are going to be produced in the body. After 5 minutes, kindly bear with me for 5 minutes, not showing the slides, okay? I will definitely share some slides and then make you understand what I was narrating, okay? This is my style of teaching. Raghunandan knows, okay? So the important compounds which are produced by different cells through the amino acids is also very important. Okay. Is it only the three important aspects of dietary amino acids? That is cellular protein synthesis, cellular biologically important compounds, and then cellular molecules, vital molecules like dopamine, epinephrine, norepinephrine, or even your purines and pyrimidines. Have you heard of purines and pyrimidines? They are the basic unit for making the nucleic acids, DNA and RNA. Trust me, purines and pyrimidines are manufactured by the body. You don't consume through diet. Even if you consume through diet, the fact says that intestine doesn't allow your dietary purine or dietary pyrimidine to go into systemic circulations and to help. Okay, but there's a debate. So purines and pyrimidines are manufactured by the body by using different amino acids for making different purines and pyrimidines. Likewise, there are more than 50,000 or lakhs of different molecules are produced from different amino acids, my dear. Take an examples of glycine. There are 10 different functions for glycine. So which means what? Textbook says 10 different functions, which means what? 10 different products can be manufactured by using lysine. Apart from making a different proteins, different peptides, 10 different molecules are produced. Each molecule is helping for a cell to survive. So I don't want you to understand at a molecular level details first. You please pay attention the concept of our diet, especially the amino acids in the diet, why we take, what are the benefits we are supposed or likely to get this out of the amino acids. Okay. Once you know very clearly, then you will be in a position to sit in the clinics to see which patients suffers from what type of problems and all. Okay. So I asked you a few minutes back, one more question. Is it the only three different aspects of functions for amino acids? Not at all. Not at all. But please trust me, these three functions are very important. The cell understands as and when it receives the amino acid, they prioritize number one for protein synthesis. Similarly, I can call all this as a number one only, but want a clarification, we are putting in a 3 lines. Okay, all the three are the same priority. What are the three priorities? Making the cellular protein, making the cellular biologically important peptides, and making different molecules in a different cells. Apart from this, your dietary amino acids, if there is excess in the cell, please make a note on this. Okay, if there is little excess in the diet, some people may consume little more than their need, little more than their immediate need. What will happen? Don't assume or presume just because you had more dietary amino acid, don't expect the cell to produce unnecessary protein, unnecessary biologically important peptides, unnecessary compounds, because cell never functions like that. Whatever they want, how much ever they want, whenever they want. 3 lines, please make a note of it. Whatever they want, whichever molecules they want, how much they needed for the cell, when at that point in time, so much only that particular molecule would be synthesized. Immediately after the function, even the biologically important peptide or protein or the compound which are produced has to be degraded. Why it's so important? Take an example of this case. Tyrosine helps to produce dopamine or epinephrine, norepinephrine. Imagine the functions of dopamine, epinephrine, norepinephrine are over for a few seconds. After that also, if these molecules are existing in the cell as a same molecule, it goes on continuously perform the same task. It's not needed. It's not needed. As and when a particular amount of neurotransmitter is needed, so much only will be produced, and then made to function. Once the functions are over, it has to be degraded completely and excreted out of the body. If not, there is going to be over functioning of that molecule unnecessarily. Even that is going to lead into disease. Example I'll give you. Serotonin. Have you heard of serotonin? What is serotonin? Again, it's a monoamine, okay, produced from tryptophan, amino acid called tryptophan. What is the biological function? Which helps to stabilize the mood. It's an happy neurotransmitter. Your level of serotonin has to be optimal. If level of serotonin goes very high or very low, both are dangerous. Very low serotonin pushes you to depression. Very high serotonin can make you maniac, hyperactive person. Both are not good. Okay. So when serotonins are produced at an optimum level, once the functions are going on slowly, it has to be maintained at least that level, or shouldn't be produced little higher level. If there is little higher level, it has to be degraded and then brought to normal level. Okay, that's what I meant. Otherwise, you will become maniac for some few minutes. You will become depression for few minutes. That's what happens in day-to-day basis for our life. Okay. Optimum amount of molecules has to be maintained. Once the functions of molecules are over, any molecule, it has to be degraded and then excretable form, it has to be made and then excreted via urines. You will see it with example. I am going to take the class now. Okay. Fine. Any doubts, major doubts if you have, you can clarify right now. Otherwise, let's start with one example of amino acids, what I was narrating. Okay, there are two questions. Are the amino acids are stored? Somebody asked the question, are the amino acids are stored? The answer is, per se, there is no organ which can store amino acid as it is. But in the case of carbohydrate, carbohydrates are stored in the form of glycogen, especially in the liver and skeletal muscle. In the case of lipids, yes, we do store lipid, how much ever is produced or how much ever is taken. Any amount of excess of lipids can be stored in the adipose tissue, but not amino acids. Amino acids are not stored, but there is a trick. Okay. The person who does exercise, he produces a bulkiness in the skeletal muscle. So the person who works out, who consumes a lot of protein, he can store the excess of amino acid in the form of skeletal muscle fiber. As and when there is a demand or need for the person, skeletal muscle protein can be degraded and supplied as an amino acids. That you must understand. But you can't say it is a storage, but it is agreed that you can degrade to certain amount of skeletal muscle when you are building up, and then again a person can eat more protein and then do exercise, then he can store. Why I'm bringing a word exercise? Without exercise, if you're consuming more dietary protein, I'm sure it's not going to become muscle protein, because the more you eat without doing any skeletal exercise, you are not going to build up the muscle fiber. Okay, that's the explanation. Last aspects, okay? The excess of amino acid, if somebody takes it, somebody takes it in the diet, what will happen to them? As I told you, don't expect all excess of amino acid to produce unnecessary protein, unnecessary biologically important peptide, unnecessary compounds for a cell. Never does like that. Supposing if you're consuming little excess. See, now youngsters likes to go for a KFC or any food, okay? You eat more protein without knowing how much you need on the particular day. But if you take it so much excess, what happens to them? You need to bother, okay? What happens? That is when your excess of amino acid is degraded, and then produced into a compound, okay, which can be eventually converted into lipids. Yes, you heard it correct. Lipids, okay. So don't be in a thought like more protein is more good. Never, never. Okay, please mark it very clearly. More than your immediate need, if you're taking through your dietary protein, eventually on the particular day itself, it's all going to be converted into lipids. Okay, the lipids means triacylglycerol by the liver, and then it will be sent through the blood circulation to the adipocyte. It is all going to be deposited in the adipocyte. That's why whether you take excess of carbohydrate or excess of protein, mark my words very clearly, excess of carbohydrate or excess of protein, everything eventually gets converted into triglyceride. This is what is normally lipids. Okay. When we say lipid, it means triglyceride. Other lipids, you have to call the correct name, cholesterol, ester, or phospholipids. But you don't have to call triglyceride. Sometimes you can just say lipid, neutral lipid or neutral fat. It means triglyceride. Okay. So excess of carbohydrate, excess of protein eventually converted into triglyceride and then stored in the adipocytes. That's why it is not advisable to consume more than immediate need of the person. More than immediate need of the person. Component of glutathione. Component of glutathione, somebody asked. I gave you the answer. Glutamic acid, cysteine, glycine. Okay. These are the three important amino acids which makes glutathione. There's a huge difference. Please understand the protein and the peptide. Proteins, more than 50 amino acid, I told you. Lesser than 50 amino acid, we call them biologically important peptides. There is a huge difference. What is this, sir? The difference is, for all protein, you need a guidance through gene. Please make a note. For all proteins in the body, whether it is a simple protein which is having only 50 amino acid or 5,000 amino acid or 10,000 amino acid. There are protein which has got 10,000 amino acid, my dear. Okay. Any protein, but every protein is guided by gene. Through their mRNA guides which amino acid to be on which place. The order of amino acid, total number of amino acid is being guided by their own specific gene. But for a peptide, there is no gene involvement. There is no gene involvement. Only the enzymes in the cytoplasm helps to synthesize the peptide. Like 9 amino acid, sequentially, first 2 amino acid will be attached by one specific enzyme. Third amino acid, another enzyme. Fourth amino acid, another enzyme. So there is no gene involvement. Peptides, please make a note of it. Peptides can be produced or synthesized with the help of enzymes in the cytoplasm itself or in any organelles. Their own enzymes helps to produce the peptide. For protein, it's totally different story because we have 1 lakh different protein. Each proteins are different from one another. How? The total number in all proteins are different. Total number is different. Even if you find some protein with the same total number, the order in which the amino acids are present is slightly different. This is how all proteins are different. How is it possible? Difference in the mRNA sequence helps to produce the different proteins. Please make a note of it. Okay. One more important point, then we will start the slides. That important point, please, it's a very important. I was organizing the answer in a 4 different line. First one is protein. Second one is biologically important compounds. Third one is molecules. Yes or no? Correct. So all these 3 aspects, nitrogen in the amino acid is present. In the protein, nitrogen is present. In the biologically important peptides, nitrogen is conserved or present. In the compound which are made from individual amino acid, even there, the nitrogens are present. Nitrogens are present. These are demarcations. All the first three priorities, nitrogens in the amino acid is present to make these molecules. But in the last aspect, if you have forgotten the last aspect, I repeat, last aspect is when you consume little excess, than the three important aspects. If a cell fulfills all the protein synthesis and biologically important compound synthesis, and then important compounds, molecules which are produced, everything is over. Still, if the cell is having little excess, that is when most of the cell will push the amino acid into plasma. That is when your plasma level of amino acid goes little higher level. So we also call amino acid pool, POOL. Okay. Amino acid pool supposed to be 1 milligrams per DL at any given point in time. If the amino acid pool level goes little higher level, that is when liver decides to take maximum amount of amino acid. Why? Liver has the ability to remove the nitrogen first. Because I told you, if you remember, all the three first important functions, nitrogen is present, which is must. But if there is little excess of amino acid, we have to manage. If the little excess of amino acid, if it is left in the plasma, it is going to increase the concentration of plasma. Osmolality of plasma going to be increased. Blood flow will be decreased. You all know the consequence. Blood pressure will be increased. And then perfusion to the organs may be reduced. And then slowly organ all will lose. Yes, even amino acid excess is such a dangerous. But that is not happening for a normal individual, you and me. Even if you take little excess, since your liver is intact, liver is good, so the all excess of amino acid is going to be taken by the liver. What they do, first they remove the nitrogen as ammonia, because we cannot excrete, even if we remove the nitrogen as a nitrogen gas, we are not having a capacity to release the nitrogen directly from our body into atmosphere. That's the reason we have an enzyme system, removes the nitrogen as amine group. Eventually, it is converted to ammonia. Even that is very dangerous. Okay. That's why almost all ammonia is carefully brought to the liver. Maximum is produced in the liver itself. Even if some organs or some cells does this function. What function? Excess of amino acid is happen to be present in an organ. If the organ removes the nitrogen, it will be removed as amine group, and then amine group is being brought by some other molecule safely through the blood to the liver. Mark my word very carefully. Safely through the blood to the liver. In the liver, all nitrogen as amine or any other form eventually converted into ammonia, especially in the mitochondria of the particular liver cell. Okay, hepatocytes, mitochondria of the hepatocyte, ammonia is formed. Mitochondria, very secured place. Your ammonia is released there. Ammonia is such a dangerous entity. I will tell you in the later how ammonia is such a dangerous. Okay, if you know the answer, please put it in the comment box. Such a dangerous molecule, ammonia. If you inhale some ammonia, I am sure you will die. At least you will get coma first. If not treated, definitely you will die. You and me will die if we are exposed to ammonia, little excess than the normal level. Okay, such a dangerous molecule. But trust me, constantly they are produced in the body, but we are not dying. Why? The simple reason is it is produced in the mitochondria of the liver cell very carefully, not leaks out. That's the meaning. Okay, it is kept inside the mitochondria of the liver cell. With that, another nonsense toxic molecule, I hope you can guess now, which is equally dangerous, nonsensical, but unfortunately we can't help it. We have to produce that in a tons of amount. That is carbon dioxide. Okay, carbon dioxide. Carbon dioxide and ammonia joins and then eventually made into urea, very safest molecule which can be excreted. This is how excess of amino acid in the system first gets removed of its nitrogen. Then what you see in the amino acid without nitrogen is almost equivalent to carbon skeletons of the carbohydrate and lipids. Please compare these aspects. That's why in the beginning, I started asking, how do you make sure what you see is amino acid, what you see is carbohydrate, what you see is lipids? It's very important. Okay. Carbohydrates are carbon number, water number should be equal. Carbon number and water number should be equal. H2O, you count them and then separately put it. Carbon alone you count. Carbon plus H2O should be equal. Carbohydrate. That's why the name came carbohydrate. Hydrate means water. Okay. For lipids, you will have to see one alcohol and one acids. Always the acid is fatty acid. Alcohol, most of the places it's glycerol. So glycerol plus fatty acid esterified. All lipids are ester. Okay. I am not here to discuss about the lipid. I'm just giving the brief idea. The first question what I asked. Similarly, for amino acids, I told you one single carbon, that is alpha carbon, should have carboxylic group, amino group. The same carbon is attached with carboxylic as well as amine group. If you see this kind of structure arrangement, you blindly call them as amino acids. No doubt here. But now, when you remove the alpha carbon atom amine group, rest everything is C, 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 H, O. That's all. It is almost comparable with carbohydrate or lipids. Now you would have got the idea. Once you remove the amine group or nitrogen group from amino acid, the cell see them as carbohydrate or lipids. How beautiful it is. Then those enzymes like glycolytic enzyme or TCA cycle enzyme can take the amino acid. Now you don't have to call amino acid the amino acid which is devoid of nitrogen. The amino acid which is devoid of nitrogen. Safely you can call carbon skeleton. Carbon skeleton. Why we call carbon skeleton? Just to make clarity, carbon with hydrogen and oxygen, those molecules grouped as carbon skeleton. This is how the excess of amino acid is managed in the body. What will happen to the carbon skeleton? What will happen to the carbon skeleton? If the person needs the energy, ATP, then these amino acid which becomes the carbon skeleton may produce ATP, If the person energy levels are very high, already he has got ATP and then completed all the task of ATP, he doesn't need much of ATP. But at that point in time, he also has excess of amino acid. Imagine excess of amino acid. He doesn't need to produce ATP out of the excess of amino acid. That is when the excess of amino acid gives away the nitrogen. Always the excess of amino acid gives away the nitrogen. But if there is a demand of energy, it goes to energetic pathway. Immediately energy can be produced. This is how the excess of amino acid can be utilized. But if that demand is not there, trust me, that is when the excess of amino acid, which become carbon skeleton, goes into lipid synthesis, triglyceride synthesis. This is how triglycerides are made. So at least if you are consuming the food, see to that you're going to be used immediately. How do we know? How do you make sure? It is only hungry cycle. Okay. You have to know how much is your satiety, how much you need for immediate usage. Okay. Okay, let me start the slide with one example of amino acid at least for half an hour and then we will wind up. Okay. Any questions you can put up in the group? Sorry. One minute.
Sir, I think you have stopped sharing, sir.
Yeah, I'm just trying now. Can you see the slide now?
Yes, sir. Yes, sir. We can see.
Okay, thank you. So far, I laid down the concept. Okay, that is what is very important. Okay, facts are not important. You have to learn in context to the human life on everyday basis. Okay, that is what I was helping. So you have to spend some time with that template, what I was narrating today. As and when you take 20 different amino acids in any protein, once it digested, 20 different amino acids will be formed in the intestine. Yes or no? Absolutely yes. Then what happens to the 20 different amino acids? They travel through the portal system to the liver. Whatever liver wants, they use them. Little excess will be there. Definitely you would have had at least 100 grams of protein or 200 grams of protein. Millions to trillions of amino acids would have formed in the digestion process. Okay, don't assume all the thing will be consumed by the liver alone. So liver takes only whatever they need. Rest everything has to go through the systemic circulation to each and every organs. When I say each and every organs, each and every cells. Then I gave you the answer. What is the fate of these individual amino acid in every cells? The fates are different protein for the individual cell, different peptides for the individual cell, biologically important peptide, different compound from different amino acid for the individual cell. This is how you have to assimilate. You have to imagine when you take, digestion happens, amino acids are released, absorb, goes through the blood, through systemic circulation, through their receptors in the cell. Every amino acid is entering into every cell. Then what happens? Inside the cell, whatever protein has to be manufactured, these amino acid contributes for manufacturing of the protein or synthesis of protein, you can use the word. Apart from that, if a cell needs any biologically important peptide, these dietary amino acids which went in the cell may contribute for synthesis of biologically important peptides. Apart from this, if a cell needs some vital molecules like serotonin, melatonin, or thyroid hormones, or dopamine, or norepinephrine, epinephrine, these amino acids are going to contribute for thousands and thousands of products like this, according to the cell. After all these functions are fulfilled, still if the person is having little excess of amino acid, that is when I told you the last event, giving away the nitrogen group first. Okay, there are specific enzyme which comes and cuts off the nitrogen as amine group. Then what you see is a carbon skeleton. That carbon skeleton is eventually managed by different enzyme to get into a compound which can be used in the glycolytic pathway or TCA cycle. That's it. So simple it is. But the textbook will not give you this idea. Okay, you should ask question, why I'm taking this food? Any vitamins also, any minerals also. First question you should ask, why the hell I take these vitamins? Where in the body these vitamins are going to be useful? Why the hell I take the minerals, each and every minerals? In what way it is contributing by my life? If you ask this, I'm telling you, my dear, you will all become a wonderful resource to the country. Okay? Please learn to ask questions. Why? Okay. Just I will rush through with 20 minutes maximum because now my concept laying down is very clear, I believe. I'm sure I would have made that. Okay. If that is true, then what you're going to see is an example. Very simple. It's an example. Okay. I am going to take one particular amino acid as an example and then try to see how this amino acid, when it enters into any cell, what way the cells are going to manage this. Likewise, I want you to learn all the other 19 amino acids. If you find difficulties, ask your boss, Parthiban, or your Dr. Raghu. Okay, through them, you may reach me, then I will help it. Okay, let's start with an individual amino acids. Okay, before you learn, I lay down the rules. Okay, whether the amino acid, even if you don't take through diet, whether it can be produced in the body or not. First, you should ask this question. What is this? Which amino acid you're going to learn now? Ask one question, whether the amino acid is manufactured in the body without taking through diet, which means a non-amino acid source. Okay, some carbohydrate or carbohydrate related compound maybe is used to produce amino acids in the body. Those are called non-essential amino acids. That's why the name came, non-essential amino acids. Whether the amino acid is manufactured in the body. If so, first learn the pathway through which the amino acid is produced. Okay, synthesis of the particular amino acid, you learn that. And then immediately you may learn, or in the end, you may learn how, when it is in excess, how it is degraded, and then how it is excreted out of the body. Because even amino acids, you may wonder, it's good. As I told you, it's not so good to keep in the plasma. Okay, it has to be degraded and then stored if possible, or degraded to get an energy, and then whatever compound it is producing as a debris, it has to be excreted out. Okay, we have to learn that. And then please learn what are the important products formed from the amino acids, including biologically important peptides. Now, I miss the protein synthesis. Why? Because it doesn't make a sense. One amino acid, in which protein is present, I'm sure you will go away from the course. Okay, if anybody asks you to learn all the amino acids in which protein. Okay, there is some.
Sir, can you hear me, sir?
Yeah, Parthi, now I can hear you.
Okay, sir.
Any disconnect happened?
Some technical issues, sir. The Wi-Fi got disconnected.
No problem. Can you see my face?
Yes, sir. It's clear,
Okay,
sir.
I'm not able to see. Okay, no problem. Fine. Okay, what we are learning now with an example of one single amino acid, what are the aspects you have to keep in mind? Okay, first thing, learn whether the amino acid is synthesized in the body. If so, please learn the pathways. Very simple idea. What molecule is the starting molecule to synthesize that what amino acids? Okay, learn that aspects, which is very good. Okay, if an amino acid, there is no pathway if that is not synthesized in the body, just label it is an essential amino acid. Okay, essential means, please don't understand, it's very important. All students, they just translate English into English. Okay, essential means, sir, it's very important. Are baga, it's not like that. All 20 different amino acids are very essential, because now you learned every amino acid is involved in multiple protein synthesis. Similarly, every individual amino acid is involved in different product formation, including biologically important peptides and compounds, vital compounds. So there is no question of some amino acids are important, some amino acids are not important. Essential means essentially you're supposed to provide through diet, because body cannot synthesize them. Hope it is very clear. Make it very clearly. Essential means body cannot produce them. Okay. Then we have to learn about how individual amino acids, finally, if it is little excess, how it is degraded. And then whatever examples of amino acids you learn on a particular day, please learn the functions. Functions means what are the important products are formed. Through that product only that amino acids are functioning, my dear. Similarly, what are the important, biologically important peptides are formed. Through that peptide only, those amino acids are performing the functions. Finally, mind you, whether degradations or functions through the product or biologically important compound, it is all possible. It is all possible if you have an enzyme. Sir, what is this? I'm not able to understand, sir, now. If that is your question, the answer is very simple. Important compounds are produced from one amino acids. Important compounds are produced from one amino acids. But you need to have an enzyme. Otherwise, amino acid will remain as an amino acid only. Because everything conversion occurs through enzymes in the body. Yes, sir, I understood. Now, what is the problem? Some people, some babies, the newborn may fail to produce the enzyme. That is when the problem. If an enzymes are not produced, a particular amino acids will be not utilized. Not utilized means particular amino acid will not help to produce the biologically important products or biologically important peptides or protein synthesis. Then huge problem. What huge problem? Life is finished. That is when death occurs. That's what you see commonly in inborn errors of metabolism. Why do you think? What's the reason? When a baby born with 24 hours, 48 hours, most of the death is due to inborn error metabolism, especially in amino acids and carbohydrate and proteins. In very simple, the baby fails to utilize the nutrients. What? Baby fails to utilize the nutrient. Why? Because fails to produce the enzyme. How important the nutrients? I lay down first itself. Nutrients are the one which construct the cell. Nutrients are the one which helps the cell to function. So when you don't have an enzyme to utilize the nutrient for making the cell, for functioning the cell, where is the life? Okay. That's what the last line, disorders of metabolism. I'm sure you got the point. Okay. Please, if you are not learned, you can watch again and again. You will get some benefits, I am very sure. Okay. I've taken example of for learning today's topic, aromatic amino acids. Okay. More precisely, phenylalanine and tyrosine. Phenylalanine and tyrosine. Just 20 minutes we will complete. I'm not going to go very speed also. Okay. Phenylalanine and tyrosine. Okay. Take example of phenylalanine. Phenylalanine, the first line says it is an essential amino acids, which means you don't have to bother how this phenylalanine is formed in the body because there is no formation at all. It's an essential amino acid. That means you have to provide through diet. If anybody consumes without phenylalanine, they will suffer. Okay. So better to take a complete protein, which is supposed to have all amino acids. Okay. All amino acids. Better to take a complete protein, which is supposed to have all amino acids. Okay. Can you see the slide, Parthi, now? The slide is seen, Parthiban? Any student can message. Are you seeing the slide?
Yes, sir. Yes, sir.
Okay, okay. Thank you. Phenylalanine is an essential amino acid, so there is no synthesis pathway. But on the other hand, tyrosine is non-essential. Why did I say so? Look at the slide. When you consume the phenylalanine, after making its own protein synthesis, or if there is any biologically important compound, if there is little excess of phenylalanine, it is all going to be converted into tyrosine. There is one simple enzyme, which is major enzyme. Of course, it's not a simple enzyme. I have the reason for it, major enzyme. The enzyme name is phenylalanine hydroxylase. You would have heard this from +2 onwards, okay, or 10th standard onwards. Phenylalanine hydroxylase. What is the job of this phenylalanine hydroxylase? As the name says, it just adds one more hydroxyl group on the phenylalanine. Okay, phenylalanine hydroxylase. As the name says, it adds one hydroxyl group on the phenylalanine. It becomes tyrosine. So essentially, tyrosine is phenylalanine plus hydroxyl group. You can say that. Okay, what all needed for this hydroxylation process to complete? You need a molecular oxygen, You need a tetrahydrobiopterine. Tetrahydrobiopterine. And then cofactors like NADPH. Then enzyme, another enzyme called dihydrobiopterine reductase. Please learn this with a greater detail. Okay, I'm not going to explain everything. It's all very simple. You have a dihydrobiopterine, it becomes tetrahydrobiopterine. That is what is a cofactor for this enzyme phenylalanine hydroxylase. Okay, NADPH serves as a reductant or reducing equivalents. Okay, now onwards, your phenylalanine, whenever you take, whichever cells receives the phenylalanine, whatever protein needed for phenylalanine, it will go for protein synthesis. After that, it is all become tyrosine. That's all. There is no phenylalanine metabolism separately. Now onwards, you have to concentrate tyrosine. Whether your dietary tyrosine you take or you took the phenylalanine, both will be in tyrosine. I hope this is very clear. There is only one function for phenylalanine apart from protein synthesis. That is to get converted into tyrosine. Because I'm again putting the first slide. Because tyrosine is manufactured or synthesized from phenylalanine, tyrosine is called non-essential. Hope it is very clear. Okay. Now let us learn catabolism of tyrosine. I am putting it first itself. How whenever there is excess of tyrosine is present in any cell, whenever there is excess of tyrosine is present in any cell than their immediate need, how it is being managed. How it is being managed. Remember I told you, the first event to occur on the any amino acid. You know what it is? Can you type a message? See the message? Can you type a message? The first event to occur on any amino acid which is excess in the cell. You are appreciated if you know the correct answer. Okay, the answer is removal of nitrogen. Removal of nitrogen in the form of amine group. Okay, the nitrogen is removed as amine group. Okay, what is the enzyme which is needed? Transfer of amine group from an amino acid. That's why the name goes transaminases. Now you may be getting the idea. Oh, yes, sir. Somebody taught me. I heard somewhere, transaminase. Now only I'm able to appreciate the role of transaminase. Okay. Now only I'm able to appreciate the role of transaminase. Okay. For any amino acids, when you learn the catabolism, the first amino acid is transaminase. Now learn the tyrosine. What happens? Tyrosine is getting transaminated. That means amine groups are removed. It becomes hydroxyphenylpyruvate. Remember, all transaminase needs PLP as a cofactor. What is PLP? Pyridoxal phosphate. I don't know how many of you are alive. Okay, because I used to ask this question to my students always. Whenever I cross 1 hour or 1.5 hour, very few are interested. Okay, I'm sure I know that biochemistry is not that easy. Okay, that's why whatever major concept I wanted to teach, I taught first itself. Now what you're seeing is only example. Even if you don't learn through me right now, no bothering. Just see the slide, understand the concept. Excess amino acid eventually has to be catabolized. That's all. It starts with removing the amine group. Removal of amine group, transaminase. Then whatever product, don't have to buy hot, don't have to mug up. Hydroxyphenylpyruvate is formed, and then that is again hydroxylated into homogentisate. Then again it is oxidized into mallyl acetoacetate. Okay, I'm just want of time and then just getting into summary only. Okay. Mallyl acetoacetate. Now what happens to the mallyl acetoacetate? Gets isomerized to fumaryl acetoacetate. Then finally, this is what is very important. That fumaryl acetoacetate is liberated into acetoacetate fumarate. Where did you hear the term fumarate? In TCA cycle, okay? TCA cycle. So from tyrosine or phenylalanine, when it goes for degradation, fumarate will be formed. This fumaric acid, okay, may enter into TCA cycle for giving a lot of ATPs. If not, everything will be converted into acetoacetate and acetyl-CoA, everything will become acetyl-CoA. Remember, acetyl-CoA is the starting material for fatty acid synthesis, cholesterol synthesis. This is how excess of phenylalanine or tyrosine eventually produces fat. This is what you have to understand. Those who are preparing for PG entrance or any other entrance, you have to learn the details. For first year MBBS to pass the academics, at least you should know the concept. At least you should know the concept. What do I mean by concept? The amino acids, when it is in excess, how it is managed by the cell. The amino acid, when it is in excess, how it is being managed in the cell. This is what I was always calling concept and concept. Hope it is clear now. Whenever it is in excess, it has to find for oxidative pathways or it has to be stored in the body. When I say oxidative pathway, immediately if there is an energy, these amino acid will be used for energy. If not, it has to be stored. Always storage is as fat. Okay. That's all for catabolism. Now let us learn about the functions of phenylalanine and tyrosine together. Because remember, phenylalanine is always converted into tyrosine. So whatever phenylalanine is there in the body, it is functioning through tyrosine. It is functioning through tyrosine. Question. It is functioning through tyrosine. Okay. Now let's learn quickly with 5 minutes of time, all the functions of tyrosine. Okay. See the first slide. Synthesis of catecholamine. I was giving the same example while I was narrating the common concept. Okay. Tyrosine gives all the catecholamines, dopamine, norepinephrine, epinephrine. How exactly? Please see the slide and learn by yourself. Tyrosine undergoes hydroxylation. So what you get is, remember, I told you tyrosine itself is phenylalanine plus hydroxyl group. Now, on top of it, one more hydroxyl group. That means dihydroxyphenylalanine you can call. I'm sure you got the idea. Dihydroxyphenylalanine you can call. Okay. Please spend some time and then learn the meaning of each compound. Then your life will become very easy. You need to know the molecules as it is. Okay. Tyrosine and phenylalanine. What's the difference? Phenylalanine is getting one hydroxyl group become tyrosine. Now you're putting another hydroxyl group and then what you're getting is DOPA. Expand the DOPA. Dihydroxyphenylalanine. Original phenylalanine in DOPA, what you see is two hydroxyl group. That's why this is called dihydroxyphenylalanine. Now what happens? Remember, the nitrogen in any compound is preserved. Only now carbon dioxide is released. That COOH group in the alpha carbon is getting cleaved off. So removal of carbon dioxide produces amine. I don't know whether you can appreciate. Amino acid means amine group, carboxyl group. Amine group, carboxyl group. When there is no carboxyl group, the product is called amine only. Dopamine. Yes, now you got it. Okay. Dopamine undergoes oxidation to produce norepinephrine. Then undergoes methylation to become epinephrine. How do you remember this? Okay. First, there is no doubt about dopamine level. Then norepinephrine. Norepinephrine means first two letters you see there. There is NO. Okay. NO. NO, we can just blindly read no. No methyl group, that is called norepinephrine. This is how I remember. Okay. This is not the right way of learning, but there has to be some tricks to learning. NO stands for no methyl group, then it is called norepinephrine. If there is addition of methyl group, it becomes epinephrine. This is how I remember. What is the one molecule which donates the methyl group in always in biological system? It is S-adenosyl methionine. S-adenosyl methionine. Okay, you will learn in methionine amino acid metabolism. Methionine is an amino acid. In the methionine amino acid metabolism, a compound which is produced is called S-adenosyl methionine. That S-adenosyl methionine is a donor of methyl group. Wherever methyl groups are needed, there are many places. Okay, please learn today itself. What are all the compound which receives the methyl group to become one more active important products? Like that, you please learn. The second functions of thyroid hormone. First aspect is over. Second, as I told you, tyrosine helps to produce thyroid hormone. Very simple. You can see the slide. Tyrosine receives first iodine, become monoiodotyrosine, dihydrotyrosine, tetrahydrotyrosine, trihydrotyrosine, like that. Okay. So that's it about the thyroid hormone. Next and the last function, synthesis of melanin. Synthesis of melanin. Remember, melanin is a black compound which is produced on the skins. Okay. Don't confuse with melatonin. Melatonin with melanin. Melatonin is a, what is the melatonin? Do you have any idea? This all sounds the same. Okay, that's why I brought the name. Melanin and melatonin. Now what you are learning is melanin. Melanin is produced from tyrosine. Melatonin is a compound which is present in the CNS, central nervous system. Okay, it's called biological clock maintenance molecule, melatonin. Okay, melatonin is formed from tryptophan. We are not learning that. Melanin, what we are learning. Very simple, melanin. Melanin also produced from tyrosine. Same example what you've seen there. Dihydroxyphenylalanine first is formed and then one more hydroxylation, my God. Third hydroxylation, fourth hydroxylation, like that. Then you get quinone compound. Quinone compound polymerizes to produce a pigment called melanin. Pigment called melanin. This is what is giving a black appearance to the skin. The more you have the black skin, it is said to be protective from UV light. You may not get any type of skin cancer. It is believed to be till date. Okay, fine. We have come to the end stage. Okay, definitely it is an end stage because if you see the first slide, 4 lines I was putting. Okay, synthesis of amino acid, then whatever compounds are produced, then final line is abnormality, disorders in the metabolism. What is this disorders in the metabolism? As I told you, what you've seen just now, 3 important functions of tyrosine. What was needed? Enzymes, enzymes, enzymes. If any enzymes are absent, so we are not getting the idea. Why enzymes are absent? Why enzymes are not produced? Why enzymes are not working? Any answer to this question? Please message. This is a very important aspect. Why a newborn fails to produce or fails to have an enzyme which is working? There is failure in working enzyme. What do you think? Simplest reason. Okay, those who are not type, the answer is very simple. Genetic abnormality. Sir, we are not understanding. That is your case. As I told you, almost 95 to 98% of all enzymes are protein in nature. All proteins are guided by gene. First time itself, I told you. All proteins are guided by gene. If there is any abnormality, what is the meaning of abnormality? Any sequence variations, that is what we call mutations. Okay. Abnormality in the gene translates into abnormal protein. If happen to be an enzyme, then abnormal enzyme. This is how an abnormal enzyme fails to metabolize a particular amino acids or particular nutrients. This is how disorders occurs. You fail to manage the molecule and then it elevates in the cell and then abnormally managed, then abnormality occurs. This is how people die of. Okay. I don't have to explain. Okay. Please learn. First slide, phenylalanine hydroxylase enzyme I was mentioning. What was the reaction? Conversion of phenylalanine into tyrosine. If that enzyme, okay, that enzyme completely absent or Look at the center one, okay, center line, defect in the enzyme, okay. In the middle row, please learn, please see that. Phenylalanine hydroxylase absent means you can call classical phenylketonuria. Decreased phenylalanine hydroxylase means persistent hyperphenylalinemia or type 2 PKU. PKU means what? Phenylketonuria. Okay. Third classification, maturational delay of hydroxylase. So ribosome produces an enzyme because all proteins are manufactured in the ribosome. It has to go to Golgi apparatus for maturation, the enzyme to be fully functional. If there is a maturational delay, then you will have a type of transient mild hyperphenylalinemia. Okay. Or remember I was telling some of the cofactors. Okay. Dihydrobiopterin, tetrahydrobiopterin, there is an enzyme. If any of these are defective, then you will have a subtype of PKU. Okay. Remember, if you know exactly what is the defect, how do you know that? You have to go and see the enzyme activity, how much it is there, whether it is increase or decrease or maturational delay or cofactor deficiency, all you have to investigate. Finally, you can name the baby as any one of the type. But whatever type, what you're going to have, the baby will have mental retardation, seizure, eczema, mousy odor of urine, any of these symptoms. Okay, all related to neurological symptoms. Why? Why? The simple reason is, okay, I'm skipping the slides. You can read. I will share the slides definitely through Parthiban. Okay. If you want, you can have the slides. Maybe it will be recorded and
Okay, sir.
it will be available also. Separately, you want the slides? I am sure I am going to share it to Parthiban. Later, he can transfer to you all the
I
slides.
will post it in the group.
Thank you. Thank you. So you have to diagnose the first conditions, whether the baby is suffering from X enzyme or Y enzymes or cofactors and so on. Okay. Simple chemical tests are there, but these are all outdated, my dear. Okay. Just for a namesake, we teach academic purpose, you learn, and it is obsolete. Okay. It is nowhere existed. What is available right now, straight away go to the enzyme activity of the baby. You just collect one blood spot and then do the enzyme. Even that is also is obsolete nowadays. Many centers, they won't perform the enzyme. Then how do you diagnose, sir? Straight away, you have to go to the gene and see the sequence of the gene. How do you come to know whether the babies are having abnormal sequence? It is absolutely possible. Okay. You all might have heard Human Genome Project. Human Genome Project. All the genes in the human beings have been sequenced, and all the gene sequence is being available. If you pay the money, you ask any gene sequence you want, you just pay the money, you will get the sequence, normal sequence, and then try to sequence the baby who was likely to suffer. Okay, then match the gene sequence of the baby and the normal sequence. Once you know there is abnormality, then you tell the parents that you have an abnormal gene. Sir, what to do then? What to do then? Why do you have to diagnose? Are there any cure? Are there any cure? Yes, there are multiple options. Okay, it is good to know what is the real problem. Then once you know the real problem, management is little okay. 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 Treatment. Okay. Once you know the phenylalanine is not metabolized, you restrict the phenylalanine containing diet. It's absolutely possible. If not in India, at least it is all available in the Western countries, which the food which is lack of phenylalanine. Okay, you just have to give and then manage the baby. Once the baby grows, matures after 2-3 years, slowly the gene mutations may be corrected. If not, you can correct. Okay, there are a lot of ways. Gene therapy can be done. Gene therapy can be done. Or maybe editing also is possible now in the research progress. Okay, gene editing in the live patients. If there is a mutation, there is an enzyme system called CRISPR-Cas9. Okay, it's a piece of information. Advanced learner can grasp this. Okay, advanced learner can grasp this. Okay, I'm telling you, if the babies are suffering from a mutation of a particular gene, if the babies are suffering from a mutation of a particular gene, or if the adult is suffering from mutation of particular gene. So there is a futuristic approach wherein you give one enzyme as an injection. It is going to go to a particular cell, sits there and scans the error mutation and then cuts off the mutation and then puts the right nucleotide, anneals or corrects everything. Then hopefully the conditions may be reversed. It's all done in experimental model, waiting in many countries. They're waiting for the approval. Maybe it'll happen in our era, definitely. Right now, it is happening in Vellore itself. One of the institute in Vellore, which is doing on an experimental basis. Okay, that's a piece of information. Why these babies are having a symptoms like what I mentioned, all the neurological symptoms and so on. Look, this is very interesting for a physician. What is the problem? Phenylalanine is not being converted into tyrosine. If you remember the enzyme PKU, phenylketonuria, or phenylalanine hydroxylase. Okay, first slide, I don't want to go back. Please remember, first slide. Phenylalanine is converted to tyrosine. What is the enzyme? Phenylalanine hydroxylase. What are we discussing? If any of the babies who fail to produce this enzyme, what will happen? Their phenylalanine level goes very high because it is not being converted to tyrosine. If tyrosine is converted, no problem at all. Unfortunately, enzyme is not there. It is not being converted. Phenylalanine is getting accumulated. Body has one tendency. If any substance abnormal level builds up, then certain genes in the body which are never functionals for the lifetime, these compounds triggers those gene and then produces abnormal enzyme. Thereby, these compounds are metabolized abnormal way and then abnormal products are formed. I'm sure you got the point. Okay, this is again a concept. You can apply everywhere. I want to repeat again. Please learn. I don't know how many of you are sitting here. Okay, please learn this. Okay, I'll go a little slow. This concept must be understood very carefully to manage any patients. Okay, any compound which is not being metabolized. In this case, phenylalanine. Why it is not being metabolized? The normal enzymes are not produced. I told you why. Maybe mutation in the gene. That's why enzymes are not produced. Okay, problem understood. What will happen next? That phenylalanine accumulated. Why, sir? How it is accumulated? Remember, what we are talking is newborn baby. Mother is feeding the milk continuously. The milk contains phenylalanine because mother's milk has got a protein in which phenylalanine will be there. Continuously as he feeds, phenylalanine goes in, not metabolized. So the entire phenylalanine is getting accumulated. Then what happens? This compound phenylalanine, amino acid phenylalanine triggers certain gene which are not triggered usually. When it is so high, it is triggering, and then what is happening? Abnormally, phenylacetate, phenylactate, phenylacetylglutamine. So many different compounds are formed. Phenylpyruvate, phenylactate, phenylacetate. So what, sir? What's the hype in it? Phenylpyruvate, phenylacetate, phenylactate. My dear, this is not going to be simply excreted in the urine and then you can have a beautiful life. What happens here? These compounds, please remember the phenylpyruvate, lactate, acetate, these compounds at a molecular level, they look like a fatty acids. Remember whom we are talking, newborn, the growing baby. These compounds looks like a fatty acid as the growing brain, neurons uses these substrate instead of fatty acid, these substances as a fatty acid, because I told you they look like a fatty acid. Unfortunately, these abnormal compound being incorporated into making of the neurons itself. Very pathetic. That's why brain itself is manufactured with a defective molecule. That's why the babies are losing the life immediately. Hope you followed. Okay, that's the molecular level explanation. Whenever an enzyme is missing, the life is threatened. Whenever the enzymes are missing, the lives are threatened. Okay, if you are okay with that, I will share the slide, use the same concept and learn everything. Okay, if you really wanted my help, come through Partiban, then we will have one more session, one more amino acid. If you like it, then we can continue. Is that okay, Parthi?
Sure, sir. We'll have.
Okay. Because I don't think it is good to continue more than 1.5, 1.45. Okay. Already we have crossed 60 plus 45 minutes. Okay. I would like to stop there because students may not have more interest. I am having a Josh. I can continue for another 1 hour also. I
Actually,
don't
we
think
have
it's
around 200 viewers, total viewers
right
when you are
now.
taking. Yes, sir.
Oh, very good, very good, very good, very good. Okay. I stop here, I feel. Okay. Let them learn with the same concept what I was given. Okay. If they need my slide or if they need my help in the form of lecture, I am willing to extend my help anytime.
Okay.
Thank you. Thank you. Shall I stop
Thank
here?
you. Thank you so much, sir. Thank you
Thank
so
you.
much for that wonderful session, sir. And we are expecting more sessions from your side because yours was the most expected one when we were sharing.
Thank you. Thank you, Parthipan. I'm here to help the learners, definitely. Those are enthusiastic learner can approach me anytime through you. Okay, definitely I'm here to support for the learning process.
Thank you
Thank
so
you
much, sir.
all,
And
first time learners. Yeah, tell me, Pati.
no, if the viewers request, we'll share the PPT in the group, sir.
Sure, man. Sure. But I will share it to you anyway. If they want
Okay,
it,
sir.
let them take it or let
Okay,
them
sir.
see the YouTube also.
We'll do.
Relearn. If they
Yeah,
want any other,
will be posted, sir.
yeah, if they want any other metabolism like carbohydrate or lipids, if they wanted to have like this kind of ideas, okay, overview. But today you see, no, I've just given the overview of amino acid metabolism. I have not touched with individual first. I was narrating the entire metabolism as it is, why you learn, what you learn. Same way, if they find difficulties in lipid metabolism or carbohydrate metabolism or enzyme or vitamins or minerals, okay, you decide what topic to be covered. Same like this, 1 hour or 1.5 hour maximum. I will help them how to help them to learn the metabolism, what they feel like learning.
Okay, sure, sir.
Okay.
Sir, and
Thank
one
you.
last request, sir.
Yeah.
Actually, we'll be posting YouTube shorts so that we can reach even more greater audience, sir. Just it will be 15 seconds clips covering very important topics or very important points, sir.
Yeah.
So, if you can just elaborate in 15 to 20 seconds on how to approach biochemistry, because everyone are feared about biochemistry exams, sir.
Sure.
So
I
we'll
will
be
do that.
cutting that part alone and we'll be posting as reels just for
Okay.
15 seconds.
I will record and send you.
Okay, sir, sure, sir.
Okay. Or now itself you wanted.
No, anything is fine, sir. You
I
can
will.
record it and
Yeah, I
we
will
just
record
want
and
it very crisp and
sure.
short.
Sure.
Okay,
Thank you,
sir.
sir.
Thank you,
Thank
sir.
you so much. I was also like watching through the YouTube. I couldn't join by Zoom. I just cut it and was watching through
Oh,
YouTube.
Raghu, I love
Really
you. I
wonderful
love you.
session.
Okay.
I was also asking one question, sir, from my side. So
Please
can
tell
you
me.
just tell, see, actually, insulin is required. The insulin in the fed state, it is the one which is going to do all the anabolic actions, if I am right.
Correct.
So that is the one that is going to accept glycolysis. Others, all lipid genesis or all the things are going to be done by the insulin action. So
Okay.
whether this protein getting converted, amino acids, the excess of amino acids are going to get converted into a glucose, it is going to get converted to a lipid, as you told, sir. So
Yeah.
that is also going to be controlled by insulin, whether that insulin is having a role over there.
Yes. Fantastic question. Okay. To make it very simple, this is how it happens in the body. Okay. Whether you take carbohydrate or protein or lipids in your diet on a regular basis, whether it is excess or low, as and when you take diet, as and when you take the diet, the immediate response by the body is to release insulin. Please make it very clear for all the learners. Why? Because insulin has got all the message how to manage the diet. how to manage the diet, how to distribute the diet to individual organ, how to distribute the diet to individual organ, how to manage the diet. I will elaborate a little bit what I was narrating. How to manage the diet. Little bit of elaborations. When you take the carbohydrate has to be properly utilized, which means what? In the blood level of carbohydrate should be maintained normal level. Rest everything has to go to required organ for their benefit. The role of insulin is to push the glucose into cells, whichever cells needs the glucose. Not only pushing inside the cell, it is also going to be seeing whether the cells are continuously utilizing or not. Those enzymes which are needed to utilize the glucose also influenced by insulin. That means with the presence of insulin, all enzymes will be activated so that glucose will be properly managed, utilized. If you still have little glucose excess, the same insulin guides the excess glucose into go for storageable form. Same way for carbohydrate, like what you've seen, for protein as well as lipid. Please trust me. Even for lipid, the dietary lipid when you take, insulin only helps to pushes the dietary lipids into cell. Insulin only sees that the dietary lipids are utilized properly. If little excess of dietary lipids are present, only insulin guides the excess of lipid to go for storage in the adipocyte. Similarly, for protein, dietary protein, digestion into amino acid, all the amino acids right now we have seen, these amino acids will be pushed into the cell. Again, insulin has got a major role. Definitely trust me. Then, not only pushing into the cell, almost all enzymes which are managing the amino acids are all influenced by insulin. Again, little excess of amino acids in the plasma, only insulin guides through enzyme getting converted into lipids and then stores. So there is no question of without insulin, you can have at least proper life. I am damn sure this is how biochemistry of insulin plays a major role in managing the diets. Not only carbohydrate and proteins and lipids, even to larger extent, your vitamins and minerals, okay, which is not explored. I think I have answered Dr. Raghu.
Yes, sir. Yes, sir. Thank you. Thank you so much, sir.
Thank
And
you.
I think it was a really a fantastic session as Parthiban told. Thank you so much, sir. And I think your favorite question, it's time for me to ask and end the session.
Yeah,
And
please.
let me see who is going to answer that. One thing which fascinated me in my first year is this one question. Is glucose is the reason for the insulin release, or is ATP is cause of insulin release? This was the question I guess you asked me, right, sir?
Yeah, one of the Viva. Okay,
Yeah,
correct. One of the
during my
Viva in your
Viva,
first
this
day.
question was asked. I think I can open this to the forum. Those who are seeing this video can just answer. Is it insulin or is it glucose or is it ATP, which is actually making the insulin to be released by pancreas? So I think we can see the comment.
Okay. Let me give a clue to the learners. Okay. Let me give the clue. I am not going to give the answer. Please watch carefully what Dr. Raghunandan was asking. Insulin is coming out of the cell, beta cells of pancreas. Okay. It is not influenced only by glucose, only by amino acid, only by lipids. All diet is influencing. That means how this pancreatic beta cell can recognize all the three types of food. Okay. Whether that is how it works or through common molecule. I just help the learners. Okay. Can you answer? Are you getting any answer, Raghu? I am not seeing.
Now only it will be like streaming, sir. Let's see. Let them put in the comments. Let's open for the next 1 week. Let the people, those who are watching it, even after the session, let them put it in the comments. And
Okay.
probably in the next session we will meet, sir. And the
Thank you.
most important thing is, I just want the people to request, sir. I can tell for the session that is mainly not focusing just on carbohydrate, lipid, fat, but the common basic pathway. So everyone comes together in one single pathway with regulations of certain hormones which are going to play a major role. I think that is the one that is required. So carbohydrate, lipid, metabolism, everything is there in the book. But ultimately, at the end of the day, how everyone going to come together is what missing, which cannot be understood from the textbook. I think that can be made as a separate session. So whenever you're free,
Sure.
that will be like an eye opener for the people. And that is what required to be understood, I guess, even for a NEET PG or a UNICET or
Okay,
any
you mean to say an integrated pathway.
integrated basic
As
concept.
and when you take a mixed diet,
So whatever it is,
how it is managed.
how the energetics is
Yes,
coming together. So because
absolutely fascinating. I am ready to do anytime. You ask your viewers whenever they are feel like having before exam. I am ready to do that.
sure, sir. Parthiban, kindly make a note of it and just arrange this session, which is actually needed
Okay.
for understanding the biochemistry.
Sure, Raghu. Very fantastic. Very fantastic. Really
Thank you,
fantastic.
sir. Thank you so much, sir.
I'm honored. I'm honored.
Thank you.
Thank you.
Parthiban, you can just conclude and close the session.
Okay. So thank you once again for making time from your busy schedule.
Thanks.
I hope all the viewers got useful information.
Thank you so
We
much.
are hoping to meet you soon again, sir.
Sure, boss. Sure.
Thank you,
Okay,
sir.
viewers. Thank you. Goodbye.
Thanks. We'll end the meeting.
You will be closing?
Okay.